Good morning, my name is Deejesh and this is Syed, and today we are presenting a novel therapy for diffuse astrocytoma.
How do we change the slide?
It's the green arrow on the remote.
All right. So first and foremost, let's quickly go through the information necessary to understand our proposal. What is diffuse astrocytoma? It is a grade 2 glioma, and once diagnosed, the standard of treatment is radiation with chemotherapy.
There are many ways to treat cancer, and one of them involves the pro-apoptotic agents BCL2 siRNA and cisplatin, which can effectively destroy tumor cells, as you can see on the slide.
So next, what is a carbon quantum dot? It is a nanoscale semiconductor particle coated in a carbon mesh. An important property is that once it's in the living organism, it's actually non-toxic. Furthermore, there's an important property called the enhanced permeability and retention effect. Simply put, small nanoscale particles such as liposomes and micelles, and most importantly, nanoparticles, can accumulate in cancerous regions. The reason for that is because of the porous new vascular tissue created by cancer.
A study in 2013 conducted by Karthik and his team has showed that it is possible to combine carbon dots with a quinolone phototrigger for infrared laser-induced drug release, and we are attempting to integrate this into our treatment plan.
All right, so the problem. What do we hope to solve? First and foremost, once you inject, for example, a chemotherapeutic like temozolomide, it affects the entire body, so there's no spatial control. Secondly, once you ingest the chemotherapeutic, you cannot control when to activate it or not inside the body. In essence, it is a one-way ticket drug activation post-consumption.
As we have mentioned, temozolomide is the main drug used in the standard of care, and the issues with it are that it is inorganic, toxic, and expensive. However, current other organic counterparts are not very effective. Furthermore, traditional imaging technologies like MRI and PET scans are imprecise due to the fact that the contrast agent cannot sufficiently accumulate in tumor regions.
All right, so what do we hope to solve? This is our approach to the problems. We'll be approaching it as follows. We'll be injecting 2 batches of carbon quantum dots. The first involves using BCL2 siRNA attached to the carbon quantum dot. We'll be using this for imaging. Then we'll inject a second batch of carbon quantum dots with the chemotherapeutic cisplatin. This will be used for chemotherapeutics. In order to activate the carbon quantum dot, it will be released using multi-infrared laser system. Now, this technology was possible thanks to the patents by Open Waters. She's actually the one who created the $100 laptop per child program, and it was thanks to the ability to use infrared lasers to go through the entire body that we can actually activate these carbon quantum dots. We hope to solve the issues with the standard of care and induce apoptosis in cancerous regions. So how are we going to synthesize this? Conversion factors in PowerPoint wasn't the best. See, we'll be synthesizing this using microwave synthesis. So yes, you can actually make this in your kitchen. We'll be adding important signaling factors, for example, the nuclear localization signal, in order to get the necessary material into the cell, into, most importantly, the nucleus. Then we'll be attaching a quinolone phototrigger, and the important part is that that's activated by the lasers. We'll be dividing it into 2 batches. The first is BCL2 siRNA and cisplatin. Each will have 1 molecule of each. Then we'll be using centrifugation to get rid of the unattached particles. This is the data we will be collecting, and we hope to successfully synthesize the carbon quantum dot.
After synthesis of the carbon dot, we will then conduct an in vitro study with an artificial blood-brain barrier, and we will test its permeability. Afterwards, we will also measure the cell uptake of 4 cell lines, 2 of which are cancer lines, as well as their cytotoxicity. Finally, we will also collect fluorescence and activation data for the carbon dots. This is the data for our in vitro study. We hypothesize that the carbon dots will be readily taken in by the tumor, and there will be laser-induced drug release. The third component of our strategy involves an in vivo study with genetically engineered mice with diffuse astrocytoma, and then using the TREMA approach we described earlier, we will inject them with 2 batches of carbon dots for imaging and therapy. Finally, we will conduct an organ tissue sampling to measure opsonization, which is a release of the carbon dot from the body, as well as cytotoxicity. This is the data we will collect for in vivo study. And we hope to have high-resolution imaging of diffuse astrocytoma as well as minimized cytotoxicity in vivo.
All right, so let's talk about why our proposal is bound to work, hopefully. So by taking advantage of the enhanced permeability and retention effect, we can get high contrast imaging using the carbon quantum dots. There will be no imaging therapy gap because as soon as you inject a BCL2 RNA, you follow that up with the cisplatin, and you also have the option to cancel. Lastly, we have site-specific drug activation. So we hope in the future to use a mathematical model to maximize the volume of the tumor we cover, and hopefully we'll be doing that using artificial intelligence, so more physical science research is required. Thank you very much. We'd like to thank Western University and the University of Toronto and the Brain Tumor Foundation for giving us the opportunity to present. Thank you.
Thank you to all of you for listening to our presentation.
Great work, guys. Particularly like the bound to work, hopefully part. Really interesting. So can you walk us through a little bit on what's known about how artificial blood-brain barrier experiments kind of, whether they've been kind of validated with the in vivo versus in vivo?
Yeah, sure, no problem. So there's an important variable called the TEER, which is the transendothelial electrical resistance, and that's used to measure basically whether something can go through the blood-brain barrier or not. It's roughly equivalent. And let's say the brain has a high value of it. Then we could artificially create a membrane that's similar in that TEER value, and we could generate something that's very similar to the actual blood-brain barrier, but obviously it's not 100% because there are also proteins and other factors that happen to be involved as well.
So BCLX, really interesting and novel choice, because there is some when you look at the experiments, there is some apoptotic effect of that. What is that going to be part of what you're going to be measuring therapeutically, or is the goal purely as a kind of imaging targeting piece?
So as far as data collection is concerned, we hope to simply be using imaging to monitor the brain tumor size over time, because we believe the most effective way to deal with, this is obviously once we've gone through and actually applied the technology, the best case scenario would be to have multiple treatments over time to reduce tumor size so that the patient can live on their regular life without having something attached to them.
I also like the bound to work, hopefully, although I'm not sure I can use it with my patients before surgery. And I also admire that you can get U of T and Western to work together. That's another kudo. I think last time we asked you this simple question. If you had to summarize your project in 1 or 2 sentences, what would it be again?
We would inject carbon dots into patients with diffuse astrocytoma, and then we would image the tumor regions, and then inject further carbon dots for the therapy.
Okay. Thank you. A question I have for you just pertains to the experience a patient would have with this. So the carbon dots would be injected, or they'd be inserted by the surgeon at the time of a tumor resection? How would that work?
So carbon dots, the best thing about them is that they can cross the blood-brain barrier and get into the nucleus. So we can simply just inject them into the blood, and then they'll make their way to basically any cancer in the entire body.
So when do you apply the laser?
Yes, so we'll be applying the laser after we inject the carbon quantum dot. So we'll be using imaging to figure out where it actually is. And then we'll point the laser using a multi-laser system, for example, the LAPS laser system. And we'll be basically pinpointing it and then spamming the laser button and hopefully
So the
killing
laser
tumor.
penetrates the skull.
Yes, we'll be using something called holography because infrared lasers can only go through 3 centimeters thick worth of tissue. However, using the technology of open waters, we can apply this holography so that it can go through the entire body and back. And this is how we'll be actually getting to the carbon quantum dots, activating them, and releasing the therapeutics.
Thank you, Team 4.